This section will discuss regulation of enzyme activity by different mechanisms. So the learning goal is to describe the role of allosteric enzymes, feedback control, and covalent modifications in regulating enzyme activity. Having the knowledge of enzyme activity, now we will see more about this enzyme catalyzed mechanism. So this mechanism can be enhanced by another process called enzyme regulation. So what happens here is that the rate of enzyme catalyzed reactions are controlled by a certain type of enzymes called regulatory enzymes. So these regulatory enzymes can work either to increase the reaction rate when more of a particular substance is needed, or it can also decrease the reaction rate if the reaction need to be stopped or when that substance is not needed anymore. So likewise, the regulatory enzymes can work to enhance the reaction rate or to decrease the reaction rate. Now, this enzyme activity is regulated by three different mechanisms. The first one is called allosteric enzyme involvement, or the second one is called as the feedback control, and the third one is called as the covalent modifications. So in this class, you need to understand what are these mechanisms and how these are regulated using one by one. Now we'll first see what is the role of these allosteric enzymes and how do they regulate the enzymatic reaction. Now these allosteric enzymes can bind with a regulatory molecule at the allosteric site. So here you see an example. So this is the enzyme molecule. Now this enzyme molecule has the active site as usual. In the allosteric enzymes, there is another site called allosteric site in addition to the active site. This allosteric site will bind a different molecule called a regulator molecule. Okay, so this is not the substrate. This is a different molecule called regulator molecule. So this regulator molecule just play its role to control the reaction as needed. Now, this allosteric site can change the shape of the enzyme. Okay, so once this the regulator molecule bound to the allosteric site, now you see what happens once the regulator is bound to the enzyme. Now the active site shape was changed, right? The active site was changed. So that is the exact function of these regulators. Once the active site was changed, now it will just attach or connect, bind with the substrate to the active site. So once this allosteric enzyme is bound to the regulator molecule, they can be either work as a positive allosteric regulation or negative allosteric regulation. Okay. Now, what happens in a positive allosteric regulation? So, in positive allosteric regulation, this regulator molecule bound enzyme complex can work efficiently to bind more substrates and to form products. Okay, so that is called as a positive control or positive regulator, and like that it will increase the reaction rate. And sometimes these allosteric enzymes can work as negative regulator. And this is the regulator molecule bind to the allosteric site, and this is the active site. So once it bound to the enzyme, complex of the regulator molecule and the enzyme has changed the active site shape in such a way that the substrate will not be able to bind there anymore. So it has negatively affect the substrate binding. Okay, remember in the previous case, the change of the active site actually positively affected to bind more substrates and to make more products. But here it's the opposite process happens. Binding of the regulator molecule to the allosteric site has hindered or disrupt the binding of the substrate to the active site. Now, because the active site shape is now different. So the negative regulator changes the shape of the active site to prevent the proper binding of the substrate. Now, if the substrate doesn't bind to the enzyme, there is no enzyme substrate complex and there is no catalysis happens. It will decrease the rate of the catalyzed reaction. So this is the negative allosteric regulation. So what you need to remember is that allosteric enzymes will have a different site called allosteric site apart from the active site. Okay, active site is still there, but allosteric site is in a different region of the same enzyme, and it can work in as a positive allosteric um, regulator in some cases, and some enzymes will work as negative allosteric regulators. Um, So they will just control the binding of the substrate to the enzyme. Okay. Now we will see the second mechanism, feedback control. So to explain this, I'm using this diagram here. So we'll try to understand this diagram first. Okay. So the letter S is showing here the substrate, and this is the substrate molecule. And the enzyme is showing as E1 here, the initial enzyme. Now the substrate will bind to the enzyme, and there is an allosteric site in these enzymes because these are regulatory enzymes, right? And then once the enzyme substrate is formed, they will make the products. So the E2 is the enzyme intermediate product complex, and E3 is another intermediate enzyme product complex, and that will form the final product, which is showing in this maroon color circles. So this is the final product. Now, as the reaction progresses, if this reaction need to be stopped, or if the reaction rate need to be decreased when there are more products, okay, when there are excess amount of end products, uh, it's normal for the reaction rate to decrease. Now, this end product itself will work as a regulator molecule in this reaction. How? So these end product molecules will go and bind with the allosteric site of the enzyme. Now, what happens when it's bound to the allosteric site? The enzyme's uh, shape is now going to be changed. Okay, so when the enzyme's active site, now active site has completely different shape. So the substrate is not going to bind to the active site because it has to fit. The shapes has to be complementary. So no further substrates will bind to the enzyme. So the reaction rate will decrease. So this is how these end products will just regulate the rate of the reaction. So this is called as feedback control. So there are two mechanisms. When the end product level is high, like this, the end product itself will go and bind with the to the enzyme allosteric site, and then substrate can't bind any. The enzyme can't bind any other substrates furthermore, so the reaction will stop. Now, in the other way, so if the end product molecules are smaller, so if it need more end products to form, then the regulator will dissociate. The regulator molecule will dissociate from the allosteric site of the enzyme, and then once it dissociates, this enzyme will come back to its normal shape, allowing substrate molecules to bind again. So that will form these intermediate products and to form the final product. So it can work in both ways, positive controlling and negative controlling. So since the product molecules itself is regulating this enzyme, this is called as the feedback control. The third type of enzyme regulation is the covalent modification. So the enzymes can undergo this covalent modification, which means they can make covalent bonds to another functional group um, to change its shape and its structure. As a result, this enzymatic activity can be controlled. Now, uh, this type of covalent modifications are reversible, which means they can actually go back to the initial original enzyme after releasing the functional group. Now, there are type of enzymes called zymogens or proenzymes. Sometimes they are called zymogens or proenzymes. They are usually produced in their inactive form, which means they are not working as an enzyme in its inactive form, and they will synthesize in a different certain place, and they can activate at a later time when they are needed for a certain site. We'll see some examples of these zymogens or proenzymes. For example, chymotrypsinogen produced in pancreas and pepsinogen produced in gastric chief cells. They will stay there in inactive form. So these are inactive enzymes. Chymotrypsinogen and pepsinogen are inactive enzymes, and they will activate at different places than where they produced. And the active form is called as chymotrypsin and pepsin, respectively. Chymotrypsinogen, pepsinogen, trypsinogen, these are some digestive enzymes. And fibrinogen, prothrombin, so these are actually blood clotting enzymes. Blood clotting enzymes that are produced in liver but activated on damaged tissues. So once there is a tissue damage, these molecules, inactive enzymes, will just transport it to the site, and then it will convert to the active enzyme called fibrin, thrombin. So they will just go accumulated in the damaged tissue and worked in the blood clotting mechanism. So just like that these zymogens are inactive enzymes, and they will undergo some covalent modifications to make the active form and activated in a different place. Phosphorylation is another type of covalent modification. So in this process, an addition of a phosphate group or a removal of a phosphate group can occur from an enzyme. So an inactive enzyme undergoes covalent modification in which an enzyme can be activated by the addition of a phosphate group, or an active enzyme can be deactivated by removal of a phosphate group. Two examples are shown here. An inactive enzyme can convert to an active enzyme by adding a phosphate group. And in this diagram here, it's showing a removal of a phosphate group. This Pi is showing the phosphate group attached to the inactive enzyme. And the phosphatase will catalyze this conversion. Now let's do a learning check. Indicate whether the following statements describe enzyme regulation by an allosteric enzyme, a zymogen, or covalent modification. So by reading the sentences, can you identify what type of regulation is that? An end product attaches to the regulatory site of the first enzyme in the reaction sequence. end product attaches to the regulatory site of the first enzyme. So if the end product is attaching, so that should be an end product attaches to the regulatory site of the first enzyme in the reaction sequence. So if there is a regulatory site in the enzyme itself, and if the end product is attaching there, then that should be an allosteric enzyme. And proinsulin forms in the pancreas. So these proenzymes are called zymogen. So it is a zymogen. And phosphorylase kinase deactivates pyruvate dehydrogenase. So the phosphorylase kinase, by the name itself, we can realize this enzyme already has a phosphate group. So if there is a phosphate group attached enzyme, then that has to be a covalent modification. That is an example for a covalent modification. And this enzyme can deactivate this particular enzyme. So that's an example for an enzyme regulation by covalent modification. So these are the answers. Okay, now let's go to the last topic of this chapter for this class, enzyme inhibition. So we will describe the type of inhibition. So enzyme activity can be inhibited by different methods. So that's what we are going to discuss here. We will describe what is reversible and irreversible inhibition and what is competitive and non-competitive inhibition. So first we will see what are these inhibitors. Now we have the idea of how an enzyme will increase the rate of the reaction by making the enzyme substrate complex to make the enzyme product complex and then to release the products. So there are some molecules called inhibitors, and they will just lead to decrease this catalytic activity, or they will negatively affect this catalytic activity. So how they can just prevent the substrate to just bind to the active site of the enzyme. So and there are two types of inhibitors called reversible inhibitors and irreversible inhibitors. Okay. So remember, we are talking about different classes of inhibitors or different types of inhibitors in this section. It's very important for you to just draw this as a diagram. So inhibitors can basically divide into two classes called reversible, like reversible inhibitors and irreversible inhibitors. Okay. Now let's say what are the reversible inhibition? The first class. Okay, we are in the first class. Now, the reversible inhibitors will cause a loss of enzyme activity that can be restored. So this is important. From the name itself, it says something that we can restore or we can go back to the original state of the enzyme mechanism. Okay, so that is good thing. But still, it is losing the enzyme activity. And so there are different ways that reversible inhibitors will work, but good thing is they do not form covalent bonds with the enzyme. Remember, we are talking about these inhibitor molecules, okay? Don't get confused with the functional groups that we discussed in the enzyme regulation before. So these are some problematic molecules, inhibitors, problematic molecules for a given enzyme reaction. So the here the reversible inhibitors can act in different ways, but they do not form covalent bonds with the enzyme. These covalent bonds are usually strong bonds, right? So since they are not forming strong bonds, then that is why this will be reversible. That's something that we can reverse. Now, reversible inhibition again can divide into two classes. Okay, one is competitive inhibitors, and the second one is called non-competitive inhibitors. The competitive inhibitors, what is it competing for? They are competing for the active site of the enzyme. So what else is going to bind to the active site? The substrate, right? The substrate is the one that is allocated for the active site. Now these inhibitors will just compete for the same active site. Okay, that's why we are saying it's competitive. And there's another type called non-competitive inhibitors. They will not bind to the active site, but to another site of the enzyme. Okay, not to the active site. So it will bind to another site of the enzyme. So both of them will just bind to the enzyme. Competitive inhibitors will compete to the active site, and non-competitive inhibitors do not compete to the active site but will bind to a different site or different region of the same enzyme. And both of them are called reversible inhibition, which means we can enzyme activity can be restored. Now we will see what is this competitive inhibitors. Okay. Now we said the competitive inhibitors will compete to the active site. So in order for them to compete to the active site, that means they can bind to the active site just like the substrate. Now, in order for them to bind to the active site, they need to have a similar chemical structure and polarity. So this is very important, right? That's how they will just compete with the substrate. They will have a chemical structure and polarity similar to the substrate. So it will then compete with the substrate for the active site. Now you see here the active site of the enzyme is here, and the substrate will just bind to that. And then this is kind of an induced fit. We can explain it in the induced fit because substrate, once it's bound, it will form the enzyme substrate complex. And that will just convert it to the enzyme product complex, and then the products will release. So this is what generally happens in the enzymatic reaction. Now, when there is an inhibitor, which is the competitive inhibitor, okay, so this is the competitive inhibitor. When the competitive inhibitor is present, then both of them will just try to go and bind to the same active site. And once this inhibitor is bound to the active site, it will prevent the substrate to bind to the active site, and then this complex will not form, right? Rather, it will have this enzyme inhibitor complex, EI complex. We don't need this EI complex because it will not make the products, or it will not convert to the enzyme product complex. So this is what happens in the presence of a competitive inhibition. Now, one thing that we can overcome this inhibition process is by increasing the substrate concentration, so it can make more of ES rather than EI complexes. Okay, so the effect can reversed by increasing substrate concentration. So that is the good thing. Increasing substrate concentration will overcome competitive inhibition process. Okay, so all of them, these are very important for you to note down. Okay, they will have the same or similar chemical structure and polarity, and both of this competitive inhibitor and the substrate both will compete to the same active site. Number 2 and number 3, The effect can be reversed by increasing substrate concentration. If there are more substrate concentration, there's less chance for inhibitors to bind to the active site. So then we can make the reaction happen. Like we can make it possible for the reaction to happen. Okay, so that's about competitive inhibitors. The second class of inhibitors was non-competitive inhibitors. So remember, the competitive and non-competitive both uh, belongs to the reversible inhibition. Okay. Now, what is the difference between non-competitive from the competitive inhibitors? These non-competitive inhibitors, as the name implies, it doesn't compete with the active site, so they will have a structure very different from the substrate because it doesn't need to have the same or similar structure because it will go and bind to a different site now. Okay, the inhibitor will go and bind to a different site. The binding of this non-competitive inhibitor to the enzyme, even though it's different, that will just change the shape of the complete enzyme. So that will prevent from binding the substrate to the enzyme. And it cannot have its effect reversed by adding more substrates. So adding more substrates now is not an option to overcome the inhibition process. So that's what happened in non-competitive inhibition. So you need to memorize the differences, how the competitive inhibitors work, and what are their features that make them to bind to the active site, and how the non-competitive inhibitors work in this case, for example. And this is what you need to memorize or need to learn: irreversible inhibition. So as the name implies, these irreversible inhibitors will completely destroy the enzyme activity because these inhibitors will covalently bind with the active site or groups of amino acids that may be near the active site. So the covalent bonds are strong bonds. Once they are bond with the amino acids near the active sites, they will no longer be able to bind to the substrates because now the inhibitor will change the shape of the enzyme and the substrate will not able to bind to the active site. So there is one example here, the enzyme serine. So the serine is one R group that contains in the active site of the enzyme. This big molecule called diisopropyl fluorophosphate. So this is an example for an irreversible inhibitor. So this inhibitor can go and bind with this R group or with the CH2OH group, right? So once it forms a covalent bond, so it's very hard for this bond to break up, and the substrate will not be able to bind to this active site anymore. That is irreversible inhibition. Okay, so this is what you need to learn or need to know in this section. Summary of competitive, non-competitive, and irreversible inhibitors. So both competitive and non-competitive inhibitors are called as reversible, right? So these are reversible inhibitors. So just look at what is the shape of the inhibitor. Competitive inhibitors have a similar shape, but non-competitive does not have a similar shape, and irreversible inhibitors may or may not have a similar shape. It doesn't actually affect the shape. Irreversible inhibitors may or may not have a similar shape to the substrate. When binding to enzyme, competitive inhibitors competes for and binds to the active site. Non-competitive inhibitors binds away from the active site to change the shape of the enzyme and its activity. This is important, okay? It will bind to a different region. Irreversible inhibitors forms a covalent bond. So these are the features, specific features of these different inhibitors. I'm gonna put squares. So these will be tested in your homework quizzes and exams. So that's why I'm just putting these down here. And reversibility. So competitive inhibition can overcome by adding more substrate. So this is another important feature. Adding more substrate reverses the inhibition. And non-competitive inhibitors will not reverse by adding more substrate, but by chemical change that removes the inhibitor. Okay. And this one, irreversible, is a permanent inhibition and that cannot be reversible. So all of them are very important features. Competes for and binds at the active sites. Okay. So the ones that I put in stars are very important for you to note down and memorize. This is the summary of what we learned. Okay, I have this learning check which will help to with your homework. So find the missing word of these sentences. A blank inhibitor has a structure that is so similar to the substrate that it can bond to the enzyme just like the substrate. When it says has a structure that is so similar, quickly it should come to your mind, what is that? It should be a competitive inhibitor, right? That's why it has the same or similar structure. Competitive inhibitors. And the second one, usually a blank inhibitor forms a covalent bond with an amino acid site group within the active site, which prevents the substrate from entering the active site or prevents catalytic activity. So what is that type of inhibitor? What are the things you need to focus on? It says it forms a covalent bond with an amino acid site group within the active site, right? So what are the three types of inhibitors that we learned? Competitive inhibitors, non-competitive inhibitors, and irreversible inhibitors. So the one that forms a covalent bond has to be the irreversible inhibitors, right? Because it says within the active site. If it's within the active site, it has to be irreversible inhibitor. Non-competitive inhibitors will not bind to the active site. It will bind to a different site than the active site. And the third one, the competitive inhibitor competes with the substrate for a place on the enzyme. The same place. That place is called as the active site on the enzyme. And when the non-competitive inhibitor is bonded to the enzyme, the non-competitive inhibitor is bonded to the enzyme, the shape of what is distorted? The shape of the enzyme is distorted. Okay. And a blank inhibitor binds to a site on the enzyme that is not the active site. So remember, both the competitive and irreversible inhibitors binds to the active site or to the closer to the active site. The one that is not bind to the active site is the non-competitive. Okay, if you figure this out, then you are good with inhibitor mechanism. Okay, so let's check our answers. Competitive, irreversible, active site, enzyme, and non-competitive inhibitors. So these are the answers. Okay, so another learning check. Identify each description of an inhibitor that is either competitive or non-competitive. Increasing substrate reverses inhibition. Then that has to be competitive inhibition. It binds to the enzyme surface but not to the active site. That has to be a non-competitive. Okay, this one is competitive inhibition. inhibition, and this is non-competitive inhibition. Its structure is similar to that of structure competitive inhibition. Inhibition is not reversed by adding more substrate. Uh, so this is from competitive versus non-competitive. So that is the non-competitive inhibition. Okay, so those are the answers. Okay, okay, good. So that's the end of this lecture series. And try homework problems using the information given. Let me know if you have any other questions. Thank you.